Hi everyone, my name is Nethra Srinivasan, and my project is the effects of the onset of menopause on the transcriptomic profile of the aging female brain. First of all, to start off with the significance of the problem, there appears to be a divergence in the occurrence of neurodegenerative diseases between men and women, but the mechanisms so far are unknown. For example, for Alzheimer's disease, more than 5.5 million Americans are living with Alzheimer's disease, of whom 2/3 are elderly women. And it's interesting because there is protection against neurodegenerative disorders for younger women. Like, for example, mitochondria from younger females are protected against beta amyloid toxicity, generate less reactive oxygen species, and release less apoptogenic signals than those from males. However, the advantage is lost in the mitochondria in older females, which happens with the onset of menopause. And furthermore, current Alzheimer's treatments only target acetylcholine and glutamate to treat symptoms, but not the root cause. And since there seems to be a divergence and the discrepancy between the two genders, it's important to figure out the underlying causes to be able to better target treatments. So first of all, I'd like to figure out what is going on as the female ages and if there are any transcriptomic differences that can help us identify genes at play. And this, which can also help us pinpoint pathways that related genes may play a role in order to target treatment effectively. Next, to talk about female aging for background information. So menopause is the end of a female's menstrual cycle and fertility, in which the ovaries no longer make estrogen and progesterone. And this does not happen just in humans, it happens in most mammals as well. So in this project, the data is collected from rats. And so we will be looking at the transitioning of menopause in rats. So the first phase, which is both in humans and in rats, is premenopause, which is about 6 months for the rat, in which they have normal menstrual cycles. Then they transition on to perimenopause at about 9 months, in which the cycles become irregular, but they haven't stopped completely. So menopause at about 9 months is starts with the final menstrual cycle. And for humans, it would have stopped for about a year when we think about human menopause. And then post-menopause is at about 15 months for rats. But this is in humans when we hit the first year mark from our final menstrual cycle. From previous studies, as the brain ages in women, there's also an increased immune response. And so we are obtaining data from this study by Mishra et al., in which we have 6 groups, 6 samples each. And this is using RNA sequencing from the Rattus norvegicus, female hippocampus. And so we have 6 groups in which there is the female rat at 6 months, and it's abbreviated as Reg6M because they have a regular cycle, 6 months. Then there's 9 months with the normal menstrual cycle, or Reg9M, 9 months irregular or irregular 9M, 9 months acyclic or acyclic 9M. Then OVX at 9M, which is overoptimized rats, which means that their ovaries have been removed. And this is significant because, and it acts as a control, because OVX represents the most important clinical features of estrogen deficiency induced for modeling postmenopausal conditions. And then there's also the female rat at 16 months acyclic menstrual cycle. And so as we continue on, I found my research question and hypothesis. So firstly, understanding the genes expressed differently in the progressive stages of female aging and menopause can help us understand risks for neurodegeneration and pathways to target in order to treat neurodegeneration, such as for Alzheimer's disease in women. So a couple of questions that I had were, how does the age or the time of the onset of menopause affect the transcriptomic expression, and which pathways are expressed? Are there biomarkers that help us identify the risk for neurodegenerative disorders? How does the expression differ in overoptimized species and regular specimens, and how can that lead to insights in the onset of neurodegenerative disease? And overall, my central question is, what are the effects of the onset of menopause on the transcriptomic profile of the aging female brain? So my hypothesis is twofold. Firstly, I hypothesize that as the females enter premenopause and perimenopause, they will express less genes related to neurodegeneration as the females in menopause and postmenopause. And secondly, I hypothesized that the age and the age of the onset also have an effect on the transcriptomics of the rat. So basically, the earlier the onset of menopause, the more likely that they're going to be expressing the transcriptome for neurodegenerative disorders such as Alzheimer's disease, which we can compare with those different groups. So for my methodology, I had 6 groups to compare, and I will be using principal component analysis, differential gene expression analysis, and hierarchical clustering. And I will be using those to relate expressions to each other to draw conclusions, analyze which genes are underexpressed and which are overexpressed in 6 groups, and look at significantly expressed genes and annotate their function on David and enricher to see the pathways expressed, such as with neuroinflammation or neurodegeneration, etc. And for my results, first of all, I did preliminary PCA between the 6 groups, and I found that there was a divergence between the regular groups and then the groups that were either irregular or acyclic. And so therefore, we then grouped the regular groups and the acyclic groups and the irregular groups. And then I did PCA, and these are the PCA graphs with all of the genes. And so between regular versus acyclic, we can see that there are 2 groups here. And for regular versus irregular, it's not that they're not two specific groups because the components are very small, and they aren't filtered yet with only the significant genes. But I went ahead and I did differential analysis, and with the further PCA, so I compared the regular versus the overoptimized, and I found that they were significantly different. And so and I also use the significant genes to make these 2 PCA plots in which we can see that they're 2 discrete groups, showing that there is a very pronounced difference between the regular and the irregular versus acyclic, and that even despite the same age, there is a difference in the transcriptome when it comes to what the regular versus the postmenopausal conditions, meaning that menopause has an effect on the transcriptomic profile. So with differential expression, with further, I did the volcano plots, and I found these significant genes. And also I've got more plots for the regular versus acyclic and the regular versus irregular. And to summarize for what I got with the significant genes, the pathways that were upregulated were the regular versus irregular, and regular versus acyclic were related to immune function. And for downregulation, it was related to oxidative phosphorylation, which is linked to neurodegeneration, and related to mitochondrial dysfunction leading to neurodegeneration. And for regular versus irregular and regular versus acyclic, they actually share for downregulation linked to higher neurodegeneration, and also a higher immune response. And for the hierarchical clustering, when I did the hierarchical clustering, I also my theories were confirmed that that the 2 groups were significant. And so through the pathway analysis on Enricher, here is the visualization of the expression of the pathways, in which over here we could see it relate to immune function, and over here is for like increased neurodegeneration related to mitochondrial dysfunction and oxidative stress. Same thing over here for regular versus acyclic, a heightened immune response, and a higher neurodegeneration linked to mitochondrial stress. So to conclude for my results, I found that there was indeed an impact on the transcriptomics of the rat, especially in relation to the mitochondrial function leading to the neurodegenerative diseases such as Alzheimer's and Parkinson's disease. And I also found that there was a heightened immune response. And so this leads to the conclusion that because of the onset of menopause, this leads to the targeting of possible treatments for neurodegenerative diseases caused by oxidative stress. So this is confirmed that from this data that menopause, that the onset of menopause somehow has an effect on the oxidative stress, causing different neurodegenerative diseases like Alzheimer's disease. And it also supports the current pathologies of Alzheimer's, because for Alzheimer's, the oxidative stress that is exacerbated damages the mitochondria, which leads to cell death and even more accumulation of beta amyloid and tau proteins and neurofibrillary tangles. And in addition, it's something interesting that the oxidative stress can actually be reversed. So when there's oxidative stress, the mitochondria structure is damaged. However, researchers found that when the causes of the oxidative stress is removed, the mitochondria structure bounces back, which could lead to a possible treatment. Now, for the limitations, certain limitations is that I was not able to extract this information myself in the lab, and I had to use data that other researchers extracted from their samples, so I was not able to control the sample size or the procedures used to collect the data. Therefore, I'm dependent on their collection strategy to obtain my results. And for further research, I'd like, if I was given the opportunity to go into a lab, I would possibly see how estrogen and progesterone supplements would then affect the transcriptomics of the rat, since in menopause, the rats are estrogen and progesterone deficient. And so by testing that, that would confirm my results. And I would also like to see if this is true in humans as well. So thank you for listening to my presentation.